All right, welcome, ladies and gentlemen, to our next lesson on how the cell cycle is regulated. So, first of all, what regulates the cell cycle? There are a number of factors. There are external factors, like are there growth factors, chemicals that are released in the body to trigger growth or proliferation of certain cells, like human growth hormone? Is there cell crowding? So, if there are a lot of cells in an area, you won't need to reproduce those cells, so therefore you don't need to have the cell cycle progress. There are also internal factors, like if the cell size is very large and it's going to continue to grow, it might be a trigger that, okay, it's time to divide the cell. There are also cell cycle checkpoints, which are going to be a particular focus for this lesson, which involve these molecules called cyclins and cyclin-dependent kinases, which we're also going to talk about. So, what is a cell cycle checkpoint? You can think of it almost like a stoplight. It is going to pause the cell cycle, and it is going to determine whether certain conditions are met. If they're met, then the cell can progress through the cell cycle. If they are not met, then the cell is going to pause in the cell cycle. So these are internal controls that are going to regulate, does the cell continue to the next stage of the cell cycle? Or even, are we going to have cell reproduction happening? And depending on which checkpoint we're looking at, there are three major ones that we're going to focus on, which are called the G1 checkpoint, the G2 checkpoint, and the M checkpoint. It'll check for certain conditions before the cell is allowed to progress through the cell cycle. There are a number of molecules and a very complex signaling pathways that are involved in determining whether a cell is going to continue in the cell cycle. And these are a particular focus of research because they have a dramatic impact on human health when they get mutated. So we're going to focus specifically on cyclins and cyclin-dependent kinases, otherwise known as CDKs. So cyclins are these proteins whose concentrations are going to change throughout the cell cycle. We say they oscillate. That means they fluctuate with a regular repeating pattern. So if we were graphing this over time and you looked at the concentration of a certain cyclin, it would go up at certain parts of the cell cycle and then go down, then go up when the cell enters that part of the cycle again, and then go down. So for instance, this could be interphase, this could be mitosis, interphase, mitosis, etc. And then what will happen is this protein will be produced. It will be made by the ribosomes right as you're going to approach interphase, and the concentrations will eventually get higher. Once you reach a certain point, the cyclin will start to be degraded and its concentration will go down. This is going to be important because each cyclin is associated with a particular cyclin-dependent kinase or CDK, and when they bind together, they are going to then end up allowing the cell to progress to the next stage in the cell cycle. So there are many different cyclins. You don't need to know particular names of cyclins or which CDK they associate with. What you do need to be able to do is recognize that if you have a question that talks about a cyclin and a CDK, that they're going to interact in a particular way. So CDKs are these kinases that are only active when they're bound to a particular cyclin. So since they're kinases, remember that means that they phosphorylate other molecules. And when they phosphorylate these other molecules, often proteins, that's going to allow cells to progress past whatever cell cycle checkpoint that they're approaching. Sometimes what will happen is these CDKs, when they're active, will then target transcription factors that will activate genes that will then trigger the cell to move to the next part of the cell cycle. So let's look at an example of this happening. So a cyclin here is going to bind to a particular CDK, and that is going to make this MPF, which is mitosis promoting factor. And a mitosis promoting factor is sometimes referring to a specific molecule. Sometimes it's used a little more generally, like here. So what we're looking at here is when the MPF or CDK/cyclin complex is formed, it's going to result in some activity because it's going to activate that kinase, which is going to phosphorylate something. Notice that if we're looking at the concentrations of molecules, the CDK concentration up here stays constant. So CDKs are just produced at a constant rate and exist at a constant concentration throughout the term of the cycle of the cell. However, the concentration of cyclin is going to change. It's going to go up. So for this particular cyclin, it's going to rise steadily throughout interphase, and it's going to peak over here during mitosis. Well, when the cyclin has a higher concentration, more of it will be bound to the CDK, which means more of it will be able to phosphorylate target proteins, which is why this cyclin-dependent kinase slash cyclin complex is able to be at its maximum activity. So once it reaches some peak, that's going to force this over into mitosis. And then after that, the cyclin concentration gets decreased dramatically. And when the cyclin concentration goes down so dramatically, that MPF, that CDK cyclin complex, is no longer able to be active. So let's talk about checkpoints. Because that's what these CDKs are usually going to be mediating. So the first checkpoint we're going to talk about is the G1 checkpoint. It's also called the restriction checkpoint in certain species. So at this point, a cell is going to decide, are we going to continue on to S phase and commit to reproducing and replicating the cell, or are we going to exit the cell cycle or pause the cell cycle, or even enter G0 and stop dividing for a while? If you have growth factors present outside of the cell, that's going to promote progression to the next step. If you have DNA damage, though, or if there's cell crowding, then that would inhibit the movement past G1 checkpoint to go on to S phase. Notice here, you're looking at some cyclin CDK complexes. Again, it's not important to know which ones. They phosphorylate these proteins that end up once they're phosphorylated, they end up coming off of a gene and allow its transcription, and then those genes allow the cell cycle to progress. So it's almost like this complicated series of switches that are helping to activate certain genes. The G2 checkpoint happens towards the end of G2. And it is going to ensure that all conditions are met so a cell can successfully complete mitosis. So what's going to happen is a really big thing is to make sure that S phase completed properly and that all of the DNA was replicated. Another thing that's important is to check for any damage in DNA. So here what you're seeing, apologies, is actually a double-stranded break. That's what that DSB stands for, and I do not know why it is doing that. Okay. I don't know what's going on there. And so notice when you have these double-stranded breaks in the DNA, that's going to activate these various molecules. It's not important to know what they are. What is important to know that is that if there's DNA damage, it's going to block progression from here to here. And when it blocks that progression, it prevents entry into mitosis. So all of these molecules interacting with each other in various ways can either say, okay, go on to mitosis, or no, pause mitosis. And then these will also probably, if there's DNA damage, they'll activate DNA repair molecules. Next checkpoint is the spindle checkpoint, otherwise known as the M checkpoint. It's going to regulate whether you're going to go on to anaphase. So this happens during metaphase. Sometimes it's called the mitosis checkpoint. Sometimes it's called the metaphase checkpoint. But what it's basically checking is, are my spindle fibers attached to both chromatids? If the spindle fibers are attached to both chromatids, that is going to activate, that's basically going to, there's a complex series of inhibitions here. So this guy right here, that symbol represents inhibiting. So separase is something that allows anaphase to onset. It's basically going to separate the two sister chromatids. It's going to allow the centromere to break apart. Separase is inhibited by this molecule, securin, which is inhibited by this molecule, which is APC, which is inhibited by this molecule. Basically, when you're doing one of these diagrams, hopefully it won't be as complex as this, but you almost have to trace it backwards to see what's activating this pathway and what's inactivating this pathway. For this spindle checkpoint, if everything is connected securely, so if you have two sister chromatids, each attached to a spindle fiber on opposite ends of the cell, and they're both pulling on the sister chromatids, it will allow them to separate. That way you don't have a misallocation of chromosomes into the different cells. So you have a complete copy of the genome in each daughter cell that's produced. What happens when this process goes wrong? Well, if the cell cycle is disrupted for some reason, so if the cell division does not happen properly, what is supposed to happen is apoptosis. If the damage is too difficult to fix, if it's too catastrophic a failure, the cell is supposed to undergo programmed cell death, and that way it does not go down the other pathway, which is to then become cancerous. Cancer is dangerous to an organism. It decreases the health of an organism, and it can be fatal. So, in order to prevent that from happening, what's supposed to happen is a cell is supposed to undergo apoptosis. However, mutations happen, problems happen. So here's a pathway showing apoptosis being initiated. So, like I said, if the DNA is too damaged, if it's unable to be fixed, what will happen is you'll have this molecule right here, P53, will then activate these molecules and will inhibit these molecules. And that's going to activate all of these molecules, which will then cause apoptosis. And apoptosis, like I said, is that programmed cell death. However, if there are mutations in some of these molecules that help to regulate the cell cycle, you can have unregulated cell division, which is cancer. This is usually due to a failure of the cell cycle checkpoint, so they're not stopping the cell cycle from progressing, even though there's DNA damage. And as this happens, more and more mutations can accumulate as a cell continues to divide, even with damage. And then that can end up causing a cell to accumulate more and more of these mutations, reproduce even faster, and then certain other genetic changes tend to happen. And a lot of the mutations that are going to cause cancers are mutations in genes that control as you movement through the cell cycle. So, for example, cyclins and CDKs. When a group of cancerous cells starts reproducing quite a bit, it can form a mass called a tumor. Tumors are generally bad. They can interfere with the function of normal tissue. They can also redirect resources from healthy cells to themselves. And if they spread to other parts of the body, that can become malignant cancer. So, what are some of the genes that are often mutated in cancerous cells? There are two different classes. There's proto-oncogenes. These are things like the cyclins and the CDKs that usually allow a cell to progress through the cell cycle. If they have a mutation that makes them extra active, so they don't even check for the proper damage before they allow the cell to progress through the cell cycle, they're called an oncogene. Onco literally means cancer. And so then these cells will start moving through the cell cycle faster and faster, accumulate more mutations that are not going to be regulated by the cell cycle, and then you end up having a tumor. Okay. There's another category of genes that are involved, and I'm going to erase things so you can see. These are called tumor suppressor genes. They are usually going to pause the cell cycle when there are issues, and that's especially in response to DNA damage. But when these things get mutated, that's going to allow they're going to not negatively impact the cell cycle. They're not going to inhibit it, and so then the cells can continue to progress through the cell cycle. And so that's why they're called tumor suppressor genes, because when they function properly, they actually prevent cancers from happening. So let's look at a mutated tumor suppressor gene. This is a very commonly mutated protein called P53, and it's mutated in a very large number of human cancers. So what normally should happen is you have some sort of DNA damage that should trigger, P53 should trigger cell cycle arrest. That means pausing the cell cycle, repairing the DNA, and then restarting the cell cycle. Or if that is not possible, then it should trigger apoptosis. When p53 is mutated and damaged, it does not pause the cell cycle or it does not trigger apoptosis. The cell cycle continues regardless of whatever mutations or problems it may have, and then that cell can become cancerous. All right, that is our final lesson for Unit 4. Stay tuned.